Hello everyone, welcome to a new session on structural databases. In this session, we will study about the various kinds of structural databases, their organization and their attributes as well. We would also gain an insight about how to use information retrieval system to obtain data and the need to analyze structural data. A database. A database is a repository or collection of information that are organized in such a way that it can easily be accessed, managed and updated. In other words, database may be defined as a collection of related tables that themselves contain lists of records. To ensure that each record is unique, each of the records is given a primary key, usually in the form of an identification number. There are basically two different kinds of databases. They are primary database and secondary database. A primary database consists of data derived experimentally such as sequences and structures of biological molecules. On the other hand, secondary database contains data derived from analysis and annotations of the data present in primary databases. In today's world, the gathering and processing of data constitute a very important part of research. Especially in the field of biology, data collection plays a pivotal role. Databases for protein structure prediction. The journey from a string of amino acids to a functionally relevant protein is not easy, and predicting its structure and function is next to impossible. The problem calls for the prediction of the tertiary structure of a protein from its amino acid sequence. This is highly desirable for the following reasons. The structure of a protein can be directly correlated to its function as a biological macromolecule. Structure prediction using experimental techniques such as X-ray crystallography are time consuming and expensive in comparison to the structure determination from a primary sequence. Many proteins such as membrane proteins do not lend themselves to the structure solution. The aim of most protein structure databases is to organize and annotate the protein structures, providing the biological community access to the experimental data in a useful way. Data included in protein structure databases often includes three-dimensional coordinates as well as experimental information such as unit cell dimensions and angles for X-ray crystallography determined structures. Though most instances in this case, either proteins or a specific structure determinations of a protein also contain sequence information, and some databases even provide means for performing sequence-based queries.